Hello, my name's Tyler. I'm a postgraduate researcher at the University of Surrey in the Department of Oncology. My work involves comprehensively investigating the tumour microenvironment, known as the TME, in early non-muscle invasive bladder cancer. The aim of this is to try to manipulate this environment to improve the clinical outcomes in the patients. The TME in bladder cancer is a very complex environment filled with lots of different factors and immune cells, which can be manipulated with immunotherapies to improve the clinical outcomes in the patients. An established immunotherapy for high-grade non-muscle invasive bladder cancer is catheterized BCG vaccine. This vaccine is injected into the bladder following the resection of the bladder tumour. In upwards of 40% of patients, though, this BCG vaccine therapy fails, so there is an urgent need to improve this therapy for patients. We've been very fortunate in having strong collaborations with our clinical team over at Royal Surrey County Hospital, and through them they've been able to provide us with preserved bladder cancer tissues and also help guide the research to try and make it more clinically translatable. So this project has quite a few different avenues we're exploring. One of them is the bacterial signatures within the tumour. This is essentially the different types of bacteria that are in and around the tumour, and we're going to correlate this with the clinical outcomes of the patients. We're also exploring the overall gene expression within the tumours, and again correlating this with the bacterial expression and the clinical outcomes in the patients. So for this third part of the project, we're going to be using vector Polaris technology. This is a really powerful imaging technology which allows us to explore up to 9 different immune cell types within the same clinical sample. Using this piece of technology, we'll be able to investigate how the immune cell profiles change pre and post BCG therapy in the patient's tumour samples. Collectively, this detailed analysis will provide us with a wealth of information which we can use to help guide future therapies.